We have loaded the plasticware and reagents into the SP100. Now we will prepare and load the nanoparticles and samples. Collect the 5 nanoparticle tubes from the refrigerated Proteograph Assay Kit box. Do not uncap the nanoparticle tubes until immediately prior to loading. Do not store the nanoparticle tubes on ice. Next, we will prepare the samples, the plasma control, and the MPE control. Remove the plasma control and MPE control tubes from the refrigerator. Confirm the centrifuge has reached 4 degrees Celsius. Centrifuge 16 samples for 2 minutes. Collect the 16 Seer sample tubes from the Proteograph Assay Kit. Carefully transfer at least 250 microliters of each sample into the appropriately labeled Seer sample tube, which has a barcode label. Minimize aspirating residues, which may settle at the bottom of the tube. Avoid aspirating lipids collected at the liquid surface. Minimize bubbles forming at the surface of the samples. Record the sample tube number with the corresponding plasma sample number or other identifier. Keep the samples refrigerated or on ice until ready to use. Next, we will load the sample tubes in tube carrier 1. Remove both tube carriers from the autoload tray and place on a workbench. Uncap the sample tubes and place into positions T1 through T16 in tube carrier 1. 16 samples must be loaded for the method to proceed. Ensure that the tubes are uncapped prior to loading. Uncap the plasma control tube and load in position T17 in tube carrier 1. Uncap the MPE control tube and load in position T18 in tube carrier 1. Ensure that all samples and control tubes are fully seated into tube carrier 1. Load tube carrier 1 on the work deck. Next, we will place the nanoparticle tubes in tube carrier 2. Uncap the nanoparticle tubes and place in tube carrier 2. Refer to the ICS for correct tube locations. Ensure that the nanoparticle tubes are uncapped prior to loading. Ensure that all nanoparticle tubes are fully seated into tube carrier 2. Load tube carrier 2 on the work deck. Ensure all sample, nanoparticle, and control tubes on both carriers are uncapped and barcode labels are visible before proceeding. Select Continue on the ICS touchscreen. When the final labware check screen appears, confirm proper loading of the SP100 deck. Select Continue to move forward with the method. We have loaded the nanoparticles and samples. Next, we will start the Proteograph method.